Hello everyone. So in today's class, let's now walk through the synthesis of thyroid hormones. T3, which is triiodothyronine, and T4, thyroxine. So it is a multi-step process, beautifully orchestrated process that takes place inside the thyroid follicles. So the cells which are responsible for this are called as thyrocytes or follicular epithelial cells. So we are going to go step by step, and I want you to focus not just on the names of the enzymes and transporters, but the physiological logic behind them. So let me go to the step 1, which is the synthesis of thyroglobulin. So for better understanding, what I did is I placed myself into the thyrocyte, and on my right hand side what you can see is the blood vessel, and my left hand side what you can see the thyroid follicular lumen, we can say. So this is the basal side and this is the apical side, right? So now we will go through the entire process of thyroid hormone synthesis over here. So as we begin inside the thyrocyte, where the rough endoplasmic reticulum synthesizes the large glycoprotein called as thyroglobulin, and this thyroglobulin is then packed in the vesicles by the Golgi apparatus, and from the Golgi apparatus it is secreted by the process called as exocytosis and finally into the follicular lumen, so which is a colloid space at the center of the follicle. Now, why is thyroglobulin important? So remember that it acts like a scaffold, a kind of a carrier for the iodinated tyrosine residues that will eventually become T3 and T4, which we are going to discuss now. After the step 1, you have understood very clearly with a graphical representation. Now step 2, where we talk about the uptake of iodide. So next, what we need is the iodide, and this iodide is the raw material for the hormone synthesis. So iodide is transported from the blood into the thyrocyte by using a sodium-iodide symporter on the basolateral membrane. So this is an active transport mechanism, meaning it uses ATP to bring iodide into the cell against its concentration gradient. And the entry of this process is regulated by the thyroid stimulating hormone. So once inside, this iodide travel across the cytoplasm and finally to reach the apical membrane, where it is transported into the follicular lumen by another transporter called as Pendrin transporter. So what is the path? Blood, thyrocyte via sodium-iodide symporter and through the cytoplasm into the follicular lumen via Pendrin. This is the step 2. Now let's focus on the step 3, which is a very important step known as iodination of thyroglobulin. Now this is where the real magic happens, all thanks to the enzyme thyroid peroxidase, which sits on the apical membrane. So this TPO performs 3 major actions. First is the oxidation. It converts iodide to iodine by using hydrogen peroxide. And next step is called as organification. So iodine is covalently bound to tyrosine residues within the thyroglobulin to form MIT, which is monoiodotyrosine with one iodine. And next is called as DIT, which is diiodotyrosine with 2 iodines. And third important step is the coupling reactions. So TPO then couples DIT to DIT to form T4, which is known as thyroxine, with MIT to DIT forms T3, which is known as triiodothyronine. Now, where does this H2O2 comes from? We all know about the cycle which uses the NADPH by the NADPH oxidase, right? So it is generated by the enzyme NADPH oxidase, which catalyzes this reaction to produce hydrogen peroxide. So all of this takes place inside the follicular lumen, that is the colloid. Now you have to focus on step 4, which is the storage. So T3, T4 are synthesized, but body does not require T3, T4 immediately. So they need to be stored somewhere else. So this produced synthesized T3 and T4 remain bound to the thyroglobulin, and they are stored in the follicular lumen until the body needs them. This is one of the only hormones stored extracellularly in such large quantities. Now next is the step 5, release of T3 and T4. Now, when your body needs these hormones, when thyroid hormone is required, thyroid stimulating hormone, where it is? Thyroid stimulating hormone is produced by anterior pituitary, right? Which stimulates the thyrocyte. Thyroid stimulating hormone stimulates the thyrocyte. For what? To endocytose the iodinated thyroglobulin back into the cell, right? Now, the vesicle fuses with the lysosome and the enzyme breaks down thyroglobulin. This releases free T3 and T4 inside the thyrocyte. So, the T3, which is approximately 20%, and T4, which is approximately 80%, are then secreted into the bloodstream through MCT8 transporter known as monocarboxylase transporter 8. So this monocarboxylase transporter 8 is a protein that acts as a transporter for the thyroid hormones, particularly T3 and T4, across the cell membranes, and it is encoded by SLC16A2 gene, and it is crucial for proper thyroid hormone distribution in the body, especially in the brain. So deficiencies in MCT8 can lead to Allan-Handon-Dudley syndrome, a severe neurodevelopmental disorder. So meanwhile, the remaining MIT and DIT are deiodinated, and the iodine is recycled back into the intracellular pool, a process called as iodine salvage. So finally, we have to talk about the peripheral activation. Although more T4 is released than T3, T3 is much more biologically active. In peripheral tissues like the thyroid, pituitary, skeletal muscle, and brown fat, an enzyme called as type 2 5' deiodinase converts T4 into T3. So this is how the peripheral conversion of T4 to T3 is taking place, right? So this is what you need to know all about the thyroid hormone synthesis and its transportation.